So one technique, I've talked about two techniques till now. Photo bleaching, use it to quantify the stoichiometry. Single particle tracking allows us to measure the diffusion coefficient, and I will show later, maybe sometimes you can infer more than just the diffusion coefficient. Okay, but you can also measure the time of interaction with a slightly more immobile structure in the cell by taking advantage of the fact that the diffusion coefficient will get altered dramatically when the molecule goes from 3D diffusion to being immobile, more or less, when it binds. This technique you might have heard about is a technique that gives you dynamical information at the more nanometer scale, and it relies on the fact that the dyes or chromophores or fluorophores, when they are in the vicinity of each other, they can undergo what is called the non-radiative energy transfer. You don't need to be physically touching each other, yet they can transfer energy which has been which is present in the excited form of the donor. So if the donor has gone to the excited state, it can either drop down to the ground state or and release a photon, of course, but otherwise it can also transfer its energy, come down to the ground state, and this acceptor dye or the fluorophore can now get to the excited state, and then this dye will emit a photon. And because these two molecules are spectrally distinct, they can be detected separately on the same system, and this is commonly used in spectroscopy. But people found a way that we have sufficient signal to noise ratio that and ability to distinguish the colors on our optical setups, that you can image and identify how much emission was coming from the donor side and how much was coming from the acceptor side, and you can take advantage of that to measure the energy transfer. The good thing about the energy transfer, or the amount of energy transfer, is that it's a distance dependent function. Okay, so if you are too close to the acceptor, the donor is too close to the acceptor, almost all of the energy can get transferred to this. And if you are far away from the acceptor, then hardly any will get transferred. And this, especially in this middle regime, is more or less a linear function of the distance, giving you a very large dynamic range over which you can distinguish conformation changes in two molecules interacting with each other, or a single molecule if you have been able to place two dyes on two parts of the same protein or DNA. Okay, and this follows this sixth order distance dependence. This is a constant for a set of dyes, and with some assumptions that I'm not going to go into detail today. But you can measure distances between two chromophores if you are able to place them properly. And you can use optical setups like the microscope that you saw before. This again could be an objective type turf or a prism type turf, but emission from both these molecules, the donor as well as the acceptor, can be imaged by spectrally separating them using a dichroic mirror onto the camera, but not on top of each other. You separate them and place them next to each other. Okay, for every donor you will have an acceptor as shown in this particular system, if you have been able to label them together with the same set of pair of dyes. And you can take these corresponding images and measure their intensities. If you are looking at image like this, you can now find their respective molecules. They're low enough in density that you can figure out that there is, so for example, this set of cluster of molecules is represented in here, for example, right? So you can find each corresponding pair, and if you look at their intensities as a function of time, you will notice if they undergo conformation changes, the molecule doesn't move around, but their intensities will move in an anti-correlated manner. Okay, and this can tell you something about the dynamics at the molecule is undergoing at the nanometer scale. Okay, so the FRET basically has a distance range from about 2 nanometers to about 10 nanometers, over which it gives this linear range of energy transfer. So that can be taken advantage of. And what you can do is you can build either an equilibrium data by taking, let's say, a particular time point in space, whether it is, or you can look at the whole trajectory as a function of time to say what happened to the molecule as a function of time. So this is one kind of a trajectory that shows you that so the way you calculate your energy transfer is the amount of acceptor intensity that you are capturing by the total intensity that is captured. And there is a correction factor called the gamma correction factor that is used to correct for the fact that each of these molecules are emitting at a completely different wavelength. Their quantum yields are different, their detection efficiencies are different, so those need to be corrected for. But that can be corrected by looking at these, using these same trajectories that are observed, especially after the photo-bleaching of one of the molecules has happened. So once you have corrected for those, these trajectories basically give you an idea of the level of energy transfer that is going on. And in this case, this molecule, this one single molecule, which has both of these dyes, seems to be flipping between 3 energy states, okay, 3 energy efficiency states. This high efficiency state where the dyes are close together, most of the signal shows up in the acceptor molecule. There's an intermediate state where both of them have half and half roughly energy. So it's energy and efficiency, transfer efficiency is about 50%. And then there's this low state where most of the dye or the fluorescence is being emitted from the donor molecule. Okay, so now you're getting this dynamics at the nanometer scale, and this can be used to build histograms either from the same trajectory or for many molecules, so that you know that that's representative of the population. And then you can infer kinetics from each of these, because you can estimate the rates of how long it takes for it to go from this state to this state and vice versa, and then it can be used to extract the rate constants for going from one state to the other state. Okay, there are several experiments that I can talk about. There's a whole range of experiments that people use. Here's an experiment where stepping of a helicase protein step by step, and hence unwinding this kind of a complex, has been captured by stepwise photo bleaching, which shows up in these populations of FRET before and FRET after state, which basically means that it has gone from this particular FRET state to this particular FRET state, and that all the molecules are doing that exercise. So this kind of data can be inferred, and then you can analyze, again, remember this Poisson distribution that we had talked about, to figure out how many hidden steps are there for this kind of a single FRET change that is happening. And you can notice by my previous discussion that this one again is not a single step. Instead, there are about 3 steps hidden in the one single FRET change step that has been seen. So by inferring this kind of a non-equilibrium data, you can now start to talk about the mechanism that might be involved in the helicase moving around. Okay. Okay, here's another example. I'm going to skip this again, very similar. There's a hidden Markov based and there's a maximum likelihood based method for analyzing these trajectories, even if the number of states is extremely large. I am going to skip this. Somebody is interested in learning more about them, I'm happy to talk to them later. Okay, and there are many more complex schemes for doing FRET now that are available. You can take advantage of the fact that the energy transfer can be calculated depending on each of these schemes, and you can come up with more complicated experimental systems where you can infer not only 2 points in space, but maybe 3 or multiple points in space. So these kinds of things have also been achieved. Okay, so I had planned for questions here. That was supposed to be my halfway, but if somebody has more questions, I can take them now. or we can take the questions after that.
Can I just interrupt with the question?
Yeah, sure.
Okay, so like the individual time traces which we saw for donor and acceptor. Now,
Yes.
I just suppose if we get a trace where the amount of drop in donor is not equal to the amount of increment in acceptor. So, what
Yes.
can be the possible explanation for this stuff?
Yes. So as I said, the reason for that is several fold. The simplest reason for them is the fact that the donor and the acceptor are not the same die. Okay, so the level at which you are transferring, the amount of energy is constant, but once the energy has been transferred, the photons will come down, but the photons are getting detected by a detector which is passing through a different spectral range of the filter that you're using. It is getting detected on a detector which has a quantum yield which is different from what was for the donor, isn't it? So all that will change the ratios. So if for example, in this case, we are using a Psi3 die and a Psi5 die. Their quantum yields are supposed to be such that they give you roughly the same level of, but if I had changed it to Psi7, I would have seen much less Psi7 signal because it is so far out in the red that the detector itself is not very sensitive at that range. Okay, and this is why we rely on the anti-correlated nature of this, and we rely, if you really want to infer the exact distance changes, you have to do these corrections. The gamma correction factor has to be applied, otherwise you won't be able to estimate the real FRET efficiency. However, if you are quite confident about the states that you are inferring and you don't see anything unusual, then people take advantage of the fact that these are otherwise clear indication of certain states existing, and then you make inferences about whether we have got an intermediate state or are we looking at the same state but different state. There's one more point I would like to add. The protein that you have attached your dye to, or the DNA where the dye is attached, or the lipid which is binding to the protein, the photophysics of the dye would change when it is bound as well. Okay, the quantum yield will change because of the binding, and hence you need to be able to probe that scenario as well. When the protein is bound in that particular state, you might see a different quantum yield, and hence will give you a higher or lower signal, which might not be exactly concomitant with the, well, it will concomitant in time, but it will not be reciprocative in terms of the intensity values that you got. So all this you have to keep in mind when doing this exercise. Okay. Did I explain?
Yes. Yeah. Right. So one more thing, like suppose we are getting a trace having like beautiful anti-correlated donor and acceptor, but exactly at the photo bleaching step, suppose I am getting a single step photo bleaching for the acceptor, but two-step photo bleaching for the donor. So
then you have two donors. Then you have two donors.
But yeah, there are times like where they are like just before photo bleaching, till before photo bleaching, they are very well anti-correlated.
Well, I'll have to look at the data, then I can give you some more specific advice. In general, if you have two explicit photo bleaching steps in the in your, so whether it is a photo bleaching step or not is something that needs to be looked at. I know of dyes which have two different intensity levels. Okay. So they fluctuate between the two intensity levels, and maybe that's what you are looking at. That's one possibility, and that would explain all the things that you mentioned till now. Okay, so we can talk about this in more detail. Feel free to just email me. All right, so I'll go forward. So I wanted to show you two specific examples, go into a little more detail on how you would solve a problem of this range. So here's one example. So the questions we were asking was, how is transcription organized in a mammalian cell? And for a long time, people had talked about what is popularly known as transcription factories. The idea that RNA polymerases do not function individually on a particular gene. Instead, there are a cluster of RNA polymerases where genes can sometimes get drawn into them, and transcription happens from those regions. And hence, RNA polymerases are present in more than one at a particular place. And this had come from data where people had done immunofluorescence imaging, where they would see these brighter spots versus dimmer spots where on the rest of the nucleus of a cell. Okay, and this would be usually antibodies specifically labeling some phosphorylated forms of the RNA polymerases, and a lot of data had been generated. And so we wanted to know, yes, we knew that super resolution imaging could be done, but how do we know for sure how many RNA polymerases are there? And so this is an example of an image where if you do epifluorescence, you see this. If you do confocal or you do sheet microscopy, then you see something like this. But when you do sheet microscopy, light sheet microscopy, and this is reflected light sheet microscopy, but then what we have done is we have also selectively activated only a very thin region, which gives us very high signal to noise ratio for imaging these single molecules. And while you can't tell by looking at this, if I zoom in, you will notice that each of these clusters are indeed well separated out. Okay, so we could see the same kinds of clusters that people talked about in their images of confocal, but of course our clusters are much more well defined and they are much smaller in size. They were always less than 100 nanometers in size. And if you just take the epifluorescence image, this is what you will get. So you can clearly tell that we can do much better super resolution. The only other problem is that you have to do super resolution and to be able to do detection well, you need to be able to label the system. And this is something I'm sure somebody has talked about at a frequency or at a labeling density that allows you to detect a feature. So for example, if your labeling is very, very poor, so this is 1/16th of what it should be, then you will not be able to tell the feature any further. So you need to be able to do this labeling at a frequency which is usually twice the frequency of the spatial feature that you're trying to recognize. Okay, so that calculation has been done, and this is one of the limitations of a system that you might think about when you are working with different kinds of probes. You have two kinds of limitations. Of course, the number of photons that will come out of a particular molecule. So this is a fluorescent protein. This is an organic dye, vastly different in the number of photons they will give out. But then look at the on-off ratio. This one stays off for very, very smart fraction, a very, very large fraction of the time, and hence means you can have a very high dense structure and the molecules will not be on all the time. Whereas this one is only 500 molecules can be in this structure, and yet one molecule will always be on because of this relatively large this on-off ratio. So we went around looking for dyes which will allow us to image this very dense structure of the RNA polymerases. And then we, of course, figured out there are other dyes that can be used which stay off for very, very long periods of time. And by doing these kinds of exercises and characterizing the dyes carefully, what we knew that when a dye comes on, that's most likely is a single dye. So we can also quantify these clusters in time. So not only we are looking for clusters in space, but we are also looking for clusters in time and take advantage of that. And there are many dyes that we characterize which have these very long off times. And by doing this exercise, we can now do temporal clustering. These temporal clusters are assigned based on the idea that whenever some molecule appeared, that must have been the same molecule that kind of gave out a very large number of photons and then kind of went blank for a very long period of time. And then you can characterize these using standardized probes and to count. So take advantage of the photo bleaching experiment and quantify how many of these temporal spatial clusters that I showed you correspond to how many molecules in vitro. And when you do that exercise, now you have a way to assign the number of temporal spatial clusters to the real number of molecules. And by doing this exercise, we did this exercise for, let's say, inside the nucleus, all the RNA polymerases that are there, and our mean number came out to be 1 RNA polymerase. So we don't know whether it is because of the immunofluorescence method that people use, that any other RNA polymerase, if they are there, have been lost. But the immunofluorescence images definitely did not comprise of transcription factories. They were just a larger conglomeration of RNA polymerases, which, because of the limited resolution, were kind of visualized as so-called clusters. Okay, and then we would try and do controls, try to show that yes, whenever you get a random co-localization of 2 molecules, you can easily predict those just by random distribution of each of the RNA polymerases. So this case, the RNA polymerase has been labeled with 2 different dyes, right? And you will find that some of these dyes will co-localize once in a while, but has nothing to do with their physical interaction. It is because of the fact that at these densities, there are always going to be some which will just fall in the same so-called, even super diffraction limited spot, they will fall together. Okay, so we can show that by doing simulations that they will fall together at these densities. So it has nothing to do with a biological function, at least for us. Now, you had to take care of a few things. Okay, we had to know that we are indeed capturing most of the images. It's not as if half of them are not labeled. So you have to estimate the labeling efficiency. The way you do this is by tagging the same molecule with two of your dyes. So we had a snap tag and a halo tag. The halo tag is used to estimate how many snap tags can I label. And we can estimate that concentration by counting how many molecules are present, which carry a halo tag, but also carry a snap tag, and how many do not. So that allowed us to estimate the labeling efficiency at around 72%. So that's a good, but not extremely great system. But we are doing this, and then we ensure that we have sampled sufficient number of molecules. So you do that by continue to image and see how many new localizations you are able to get. And then you can do this fitting exercise to estimate what was the number of molecules you would have get at the end of the day if you had continued imaging for a very, very long period of time. By doing this exercise, you can estimate what is the real number of molecules and how close you are to this. And all these corrections allow you to estimate whether you have been able to image what you claimed you were imaging and how close you are to the real numbers, so making it extremely quantitative. Okay, this plot shows that most of these molecules that we did were within 100 to 200 nanometers of the Z section that we were creating. So this is an optical section that we created with our light sheet microscopy, and that so we were getting very little background from anywhere else. Okay. And remember the fact that you can take advantage of diffusion. So if you take these data in a live cell, you can distinguish the molecules based on whether these molecules stay bound for very long periods of time, or they kind of disappear very, very quickly. Okay, and these two maps are from these two categories. And you can clearly see, of course, the molecules that stay bound, they seem to be very, very well defined at certain regions. That's because they're bound to the DNA, they are going in transcription, and there are very few places where the gene's active transcription is going on. Whereas other molecules that are transiently binding to this nucleus, they will be spread much more diffuse, and you can capture this idea by just looking at these two. You can in fact map them, and what we find is some places they do indeed, there are clusters of RNA polymerases which are diffusely and transiently binding to things like this one, for example. And so it's not as if that there's absolutely no transcription factories. There are places where there exist these kinds of complexes which are pre-existing and are binding to the DNA once in a while, but most of the time they were single RNA polymerases transcribing on the genes. Okay, now I'll give you one more example before I stop, since I have maybe 5 minutes or so. So I will basically scale this problem to an even larger number of orbits. So now I said we had to quantify, and quantifying one molecule, though hard, it is possible, right? We could quantify in a single molecule. Now I'll try to quantify a very large number of molecules. Okay, the system that I'm talking about is this system of pore-forming toxins. These are bacterial toxins that bind to the, so they are produced in a water-soluble form by the bacteria. They are released by the bacteria. They could bind to the membrane of a mammalian cell, undergo large conformation changes, assemble into a ring-like structure, and then they would puncture a hole into these mammalian cells, okay, into the membranes. And these systems can be as large as sometimes hundreds of molecules. So I'm going to discuss one where we have about 12 of these molecules coming to make this ring. And the question that people have been asking themselves is, of course, how do they assemble? What's the best way to assemble these kinds of higher order structures? Because if you do sequential assembly of many of these pores, you will create a pore nicely, but it takes a lot of time to do this. If you do non-sequential assembly, where two of these molecules can combine with another two of these molecules, make four, and so on and so forth, grow in the non-sequential fashion, then they will make these pores very, very quickly, but they will also get trapped in a local so intermediate where I have created an 11 mer, but I have run out of monomers to complete it into a 12 mer anymore. Okay. The other thing has been that these are produced by bacteria. They also have a membrane. How come they do not cause disruption of those membranes? They don't puncture holes into those membranes, but they completely kill mammalian cells at very low concentrations, nanomolar concentrations, sub-nanomolar concentrations sometimes. Okay, so I will skip some of this data because some of this is already published. What we figured out is cholesterol makes a big difference. Okay, if protein receptors for at least the protein which is cytolysine A in this case seem to be not so important. The proteins are not so important, but cholesterol, if you extracted cholesterol out of them, it would basically stop the cell from getting killed. Okay, so what we do is we tag these molecules, let them bind to the membrane, but this case we are not using supported by, as we are not using cell membranes, we are using supported bilayer membranes, and then we allow them, and we basically allow them to bind to the membrane, and then we track either at low concentrations where we can look at their diffusion, or at high concentrations when they can assemble into a, and I will show you two of those examples. Let's see if I can play this movie. So here's these particles. They have been flown in solution. They will bind to the surface. As they bind to the surface, you won't see them moving around too much in 3D, but when they bind to the surface, they are moving in 2D, and that those tracks are something that we can follow. And if you just count the number of molecules as a function of time, you will find that if you had cholesterol in the membrane, then they don't seem to be binding as much as they would bind otherwise. Okay, so it looks like cholesterol did not help these proteins in binding to the membrane better. Okay, that would have been the obvious thing. Cholesterol is a receptor for the protein, and hence it binds quickly and kills the cells. But that's not what was happening. But if you look at how they move around on the cell, and this is a slightly different way of plotting the diffusional properties. So what is going on in this case is the instantaneous square displacement is plotted, which basically stands for how much did it move for very small steps. Okay, and the way to read it is, if it is on this way on the right hand side, then it is moving fast. If it is moving to the this way, then it's moving slowly. So this is not the mean square displacement, which is an average of all of the molecules, but the distribution of all the molecules moving in a particular way. If you see, initially when it binds at very, very early time points, most of the molecules are moving very, very fast. That's the red curve here. As time goes by on UPC membranes, they seem to be moving and becoming slower and slower, but a fast population remains such that they're either moving fast or they're moving slow. Okay, these two populations here. Look what happens when you have cholesterol in the membrane. You find that everything binds very, very fast, but then everything starts moving very, very slow within a few minutes of binding. Okay, you might argue, okay, this could be happening because of several things, including they coming together, and you can fit them using some other methods, and you basically find that the same kind of changes have happened. But more importantly, they are not happening because they are moving by and binding to another partner. Okay, how do we know that? You can also measure their intensities. So as a function of time, as they go from initial binding to these slow moving fractions, you will find that their intensities haven't really increased. If anything, their intensities have dropped a little. And this goes back to what Zebulina was asking. I don't find the intensities always change the way we want it to change. That's because there are other photophysical effects that would be playing a role as well. Okay, so the funny thing is, if you look at the same molecule over time, you will find that it kind of diffuses in a certain way, moving very slow. So this is again how much it has moved from its previous position. It's moving. So the blue dots are basically where it's moving very, very slow. And once in a while, it moves very far from its original place. And then it's moving some intermediate levels. They have been, of course, classified using a hidden Markov-based model. So I'm not going to tell you more about that right now. And then you can classify these different states in the same trajectory. Okay. So previously I said a molecule moves around, it will move around at a particular diffusion coefficient. Here, looks like the molecule is changing its diffusion coefficient. Why is it changing its diffusion coefficient? Because the way it is interacting with the system has changed. So we knew, and then you can extract these things from, again, doing what is called transition density plots. What is the diffusion coefficient before and what is the diffusion coefficient after when they make these changes from one particular state to another particular state. Okay, again, using a very similar hidden Markov-based model to extract the rates as well as the diffusion coefficient when they made these changes. So you extract these, and the summary of this is the fact that I have these molecules which initially bind in a certain way to the membrane would diffuse very, very fast on the membrane. We knew this from a fact that this was one of the most hydrophobic regions in this and was predicted to be the most commonly found motif to bind to the membrane. But look at this, it binds only half of the membrane, which meant that it could diffuse much faster than when the protein will completely be bound. And the changes in diffusivities that we were seeing was the changes in diffusivities as it went from this state to this state reversibly. Okay, so they would again extract these things out, and then they would move fast. And then when they would embed these things, this domain, then they would move slowly. By looking at the single molecule trajectories, we were able to extract that out. And then, of course, measure their kinetics as well without knowing what was the structural change. And then, of course, we confirmed it using several methods, which I'm going to skip for the time being. Okay, so these are, then I want to show you one more example of, so now we have gone to a stage where we have added a lot more protein so that they are no longer single molecule, they are completely capable of assembling into a structure. As time goes by, these molecules assemble. We'll see the number of spots have gone down, but the intensity of the spots has gone up. That's because they have formed these complexes. And you can measure the intensity as a function of time and extract, again, photobleaching steps for each of these kinds of complexes. And what we found is that when you had cholesterol, not only was it able to move slowly and move faster, it was also able to assemble better. Okay, how do we know this? What you see is the number of photobleaching steps that I observe for a particular scenario. I told you that I can observe as many as 12 photobleaching steps because there is a 12-mer. Of course, the fraction of 12 photobleaching steps is extremely small, though non-zero. And that's because not all the molecules are labeled. If I take the monomer, that shows mostly single photobleaching steps. If you take POPC, which is a particular lipid, it will give you some assembly. You can see that the monomeric fraction has gone down, other fractions have gone up. But if you take POPC cholesterol, monomeric fraction becomes very, very small, or at least small enough that the other fractions are long. So the assembly into higher order structures is happening better than it would have happened otherwise. Okay, so you can, doing this photobleaching analysis, you can predict this. Okay, I'll cut a long story short by telling you that you can follow these in real time. Okay, so when we measure, and some corrections have been done for predicting the exact number of particles which are in a particular form, and that's something that I can tell you more about at a later stage if you're interested. But you can estimate the exact fraction of molecules which are monomeric, exact fraction of molecules which are there. And what you notice is that assembly on the membrane doesn't proceed in a gradual fashion. Something is waiting for something to happen in these membranes, and if you ask me later, I will tell you what it is. But at some point of time, there is a very rapid drop in the monomeric and the dimeric fractions, and all the molecules very quickly assemble into these higher order structures. So some kind of a nucleation and some kind of a rearrangement in the membrane needs to happen before this process can happen. Okay, so you can capture these processes again by looking at the photobleaching as a function of time, and we do fit them to models to extract which model, which kind of a kinetic scheme do they follow. And at the end of the day, we strongly believe that now there is a sequential reversible mechanism which involves a nucleation. And I didn't get a chance to show you the data, but about a 10-fold increase because of the presence of the cholesterol, which also helps in the assembly process. Okay. Now, we don't know how the pore formation happens, but that's a story for later. I would stop here and just thank these folks. So I showed you some of the data from Satya and Pradeep, who work on the membrane pore forming toxins, and some of the data for the RNA polymerases acquired with Winston and Christoph, which was the work that we continued from Harvard. All right, thanks everyone for staying back, and sorry for overshooting the time. I can take any questions